Okay, so hi, my name is Kevin Hu. I'm a medical student here at Axel Feldmann Medicine at Mount Sinai. I worked for the MyOn lab here this summer, and this here is...
I'm Axel. I'm a high school student who also worked here at MyOn lab during the summer.
Today we're going to give you a presentation about going from the GEO to gene set libraries. Why we might want to go through some of this, it's actually, we want to look at some interesting biology regarding the transcription factors and drug perturbations. But without further ado, Axel's going to get us started by telling us what is the GEO.
So GEO is the Gene Expression Omnibus, and it's an NCBI project, much like PubMed. And it's a collection of microarray data from across the years and different types of files. So we have the nicely processed GDS files, which are curated. The terms are standardized, they're divided up into nice subsets, and they're easier to work with. But there are only about 5,000 of them. And the vast majority of the data is stored in 49,000 or so GSC files, which are not edited, have unstandardized terms, and are quite unwieldy sometimes. And these are all spread out across 2,000 different types of platforms or microarray chips. There aren't actually 2,000. Many of them are just tiny variations on standard ones, and that's what brings the number up. So, just to start off, how do we access these GeoData sets? So they have simple user access, where you just go to the website, you can look at the data sets, you can download them, you can look at the groups and the descriptions. And then afterwards, there is the Entree Utilities programmatic access API, which are just a set of URL commands. I mostly use ESearch and ESearch. So eFetch is if you have the dataset identifier number, you can do an eFetch in your URL, as is kind of seen at the bottom, except that's an eSearch. And this will give you the description for the paper, the FTP download link, the type of platform, the species, so it'll give you all the basic information regarding it in just a nice text format. Following that, there's the eSearch, which is what I'm demonstrating a short link at the bottom with, which is if you have a search term, for instance, a drug or a transcription factor or a gene or whatever you want to search, and you can do an eSearch command for that, and then that will bring up all the slightly relevant data set numbers pertaining to that. And there's also FTP dataset downloading, 1,000 dataset identifiers relating to a matrix, which is a drug. And then, continuing on, it's important when we're compiling these gene set libraries to find the right data sets. So eSearch will produce a lot of data sets, the same way Google search will produce a lot of results. But not every Google search result is what you're looking for. It just has a mention of what you're looking for at some point. And on Geo, it's actually a bit annoying because it will search, and even if your term is in the citations of the paper or anywhere, it'll bring it up as a relevant result to your eSearch. So you do end up getting a lot of files that are just not related at all to your terms. But what you can do is you can access all of the eFetch descriptions from the 49,000 GSC files and the 5,000 GDS files and compile them into an offline text file. And what this will allow you to do is you can take your eSearch results and you can just run them all quickly through your offline file, so you don't have to access millions of times, and see if the term is actually in the description. And this is a very good indicator of whether it's actually about your term or not. So continuing on, here is what the eFetch text file looks like. So you'll see that it has the identifier at the top left corner, and along the side, then we have the description. Then you'll see the organism, the type, which all the expression profiling about right pretty much, the platform, which also is the number of samples, then the FTP download link, and here is the GSC number, right here. So that's what that looks like. And then continuing on, here's the code which I use to find all of the relevant experiments. I guess it's not very interesting to look at here, but it works. And continuing on, is this really the right data? Okay, so we've done the eSearches, we've run all of the data sets which we found from the eSearches through the descriptions and seen if the terms are there, but is this already the right data? And the answer is, as usual, no. because so far it's gathered a list of experiments which have the drug in or the transcription factor in the description. But this doesn't necessarily mean that they'll compare the control states to an experimental state treated with the drug, which is what we need to generate the gene set library. And this is where we need to go in a bit more manually and just kind of toss out some things and keep the others. So I created a program which I like to call the Improver, which will prompt you, and so you call it with approved experiments of the output of the previous function, so the one which brought together the entire list of all the experiments which are slightly relevant. And then afterwards, you put just an output file which it writes to, and a tested list file which it writes to. And the tested list is where it writes what it's already tested, so if you restart it another day, it won't have to go through the same ones which you went through before. And then there are 4 commands which I'll talk about which you can answer. So it'll ask you, is it about a certain drug? And then you can answer with 1 of 4 commands that are outlined here. And that's just the code for it again. And it's going to bring up the experiment, so it's going to open the web page for it. And then it's going to ask you if it's relevant. If you type 1, you accept it. If you type 2, you reject it. If you type 3, you store it. So you put it on the side. You say, maybe it's not perfect. I can see what I can do with it later. And then you press 0 to quit and save to your output file and your tested list file. And here's just quickly the sample website which the approval will bring up for a GDS file. So it'll show you all the groups. For instance, I don't think you can see here, but it says untreated, glipizide, metformin, and it basically shows the different groups which you'll later have to identify as controlled experimental. And that's really the final part of this initial part of the process, which is to find what is the control group and what is the experimental group. In order to find differentially expressed genes, you need to know what are they differentially expressed from. And in this case, you need a control group of samples, and you need an experimental group of samples, and then you compare the expression data. In order to find this, it would be nice if you could just search in each one for the term control or the term experimental, but it always isn't. But it is very rarely the case that they label them nicely. The labels will vary from abbreviations of control, wild type, untreated. Sometimes they'll just leave them blank or things like that. So finding it automatically is difficult. I'm sure it's feasible. I didn't do it myself already, so I guess that's possibly a future thing to do, to try to get this more automatically. But for these approved experiments, what you can do is, it's another approver type thing, which will bring up all of the subset groups, and it will ask you what's part of the control group and what's part of the experimental group. So you'll see that each one of these is a subset of the initial file. So you'll see, for instance, this is the sample name, then control, breast tumor, and they'll give you some more information about it. Control, and then here's the relevant experimental group. So what I could do here is I could select either numbers 1 and 2. As my control group, I could select either the data set identifier as my control group. I can set up lists of either, so there are multiple selection methods which I put in, and you make it pretty easy to select which ones are your control groups and which ones are your experimental groups. And this one's for the GSC files, because the GSC files are getting a bit more unruly. The GDS one is based on subsets which are present. So whereas you see here that each individual one is, in fact, individual, the GDS file would put these together as a subset called control, and these together as a subset called BMS 754807. So that's the only difference between the GDS and the GSC approver is that. And now it's kind of this part.
So why do we go through this whole process of trying to find the control experimentals? Is because eventually we want to be able to define for ourselves. So what we actually want is a list of these experiments which have made comparisons under some condition of perturbation, and we want to catalog these. We are, after all, gene set librarians. So before I went on, it would also be useful, I thought, to talk a little bit about kind of the research background and what the hope is, why you want to even build a library in the first place. And so there are a few questions that I think one can actually ask once you have a library, including, basically, can we obtain useful insight by combining networks, interactive networks with functional networks? So what this means in the case of Axel's and my library is that we talk about drug perturbations, and then we look at, okay, what gene expression changes existed, and then for me, it's what gene expression changes happened in response to a transcription factor. This basically, after you have a whole library of this, you can actually extract a functional network from it. But it's not complete without a corresponding interactome network that tells you, okay, these gene expression changes correspond to actual, perhaps other physical interactions. So that's from, say, a library like the CHIA, which catalogs all these ChIP-seq experiments that characterize actual binding events, right? And so if you combine these two pieces of information, maybe you can also derive some other interesting biological insight. Other things that these libraries can be used to do is that you can talk about, this is in Neil's project, basically looking at a new way of calling genes using characteristic direction. And basically, once you have these experiments, you can use different ways to actually, you can use characteristic direction or use a t-test in order to call which ones are the differentially expressed genes. You get two different libraries, and then you can potentially validate how well your library, or how good these libraries are, basically in a downstream step where you run some other tests and analysis. And then finally, GMPs are really interesting because you can make them straight up crisp sets, which traditionally that's what people did, where you have an annotation term associated with a whole bunch of genes. Or you can actually make them into fuzzy sets, which are really interesting because you can then attach to each gene some sort of score that characterizes its degree of membership. And again, this is a lot of really cool stuff that Neil's been working on, and that's sort of how I feel like our stuff fits in. So basically, this is just the characteristics of the stuff that I found using Axel's approver, right? I had 152 total studies with 112 unique transcription factors. Just again, for context, CHIA has about 170 some now, and then ENCODE also has about 160, and then combined between them there are like 268 unique ones, so this can be grown for sure. And then this gives you a sense of the overlap between my set and CHIA and my set and ENCODE, 5327, and then the total number of target genes that were present in the entire GMT. So that's enough about kind of the end product. Let's go back to actually producing it. There we go. So gene calling. So this is basically the next software processing, data processing step. You've got all your experiments. That's great. So what you basically have is a set of accession numbers, and then you have their sort of associated experimental and control groups, control and experimental groups, and you want to actually get a useful output, which is a bunch of lists of differentially expressed genes. So the way we do that, again, is we've got 3 Python scripts, or actually 4 Python scripts that enable this to actually work. This one is sort of the wrapper script that I wrote this summer, and then the getter is by Chris Tan, and the parser is mostly by Ed, and characteristic direction implementation is by China. Yeah. So this, again, kind of gives you a sense of the code structure, basically how the processor interacts with these 2 other scripts. These are the steps. Basically, you have your experiments and the information that you got from the approver, controls and experimentals. We pass that into the processor. It passes it to the getter, which pulls the data from GEO using a URL library. And then once you have that data, it passes it on eventually to the parser, which gives you the differentially expressed genes, and finally outputs into that directory where you have all your nice gene lists set up. Again, this kind of gives you a close-up of the kind of information that you can pass into the processor. Again, it would be, here you have your accession number, here you have a cutoff PubMed ID, all these sort of highlighted in yellow is just various optional annotation information that would be different for whatever kind of GMT you're trying to actually create. This gives the transcription factor of interest, control and experimental group, and also platform, which, as Axel mentioned earlier, is somewhat important because they have all different layouts in terms of what kinds of genes they're going to be detecting. In terms of the code, you can sort of see that the main function, the business end, is the process accession list. You pass it in as a file. You can indicate whether you want to use characteristic direction when you're actually doing the gene calling or not. So that's pretty straightforward. Then, in terms of the actual getter, this is the two main functions that Chris has written, which call many other methods that are elsewhere in the script. But basically, this is what dataset identifier is passed off from the processor. And finally, once you get into the parser, there are two functions that are written. So the as parser originally could only parse through soft files, and then we kind of just modified it slightly so that it would be able to deal with series matrix files as well. And the important things here are, again, this whether or not you're going to use characteristic direction, that's going to be automatically passed in. And then, of course, because you had your accession list text file, you're going to get these control names and experimental names set up, and it's going to be able to then either do a t-test or call your characteristic direction script. So that gives you your list of genes. So once you have them, you want to actually have a GMT. And so this is the final piece of code, which I haven't shown because it's pretty simple, but it'll go through all your lists in the directory and output a library. And it's at this point that you can apply any membership functions that you want if you're going to create a fuzzy set, or in very straightforward way, make a crisp set. To give people a summary of what's actually happening in Enricher now, what's been submitted by both Axel and myself. We've got a crisp set, t-test. We've also got, I should say, fuzzy set for the t-test, and a crisp character direction, fuzzy character direction. Those are all actually in Enricher. Both up and down lists are present as long as they had enough genes in them. And then also the list length of the characteristic direction GMTs was actually determined by the length of the t-test gene lists. And this was just actually an expedient to create a tool that could be easily uploaded and used, but it's obviously something that could be looked into in terms of how you want to cut off, how you want to set these thresholds, and that's all something is interesting to potentially look at. So something that ended up taking up an inordinately long amount of time after you even get your GMT was sort of cleaning it up and converting nonsensical symbols into sensible ones. And I just thought I'd put up some resources here that I found useful. The main one being biodb.net, which you can input thousands and thousands of different symbols into using their db-find function. You don't even have to know what they are or what they mean, and it'll try to convert them to gene symbols. If it succeeds, you can keep it, convert it, and if it doesn't succeed, you can say, okay, great, I can get rid of these. Just anecdotally, my GMT ended up having about 10% of the symbols be non-standard, meaning just non-official gene. And these other resources are just great places to go. Or sorry, the NCBI MTG is just a really great place to go for anything you want in terms of up-to-date gene lists. So this sort of wraps up my portion of the talk. So there's lots of things that you can do now once you have your GMT. First of all, the sort of process that we just described, we're thinking it could be certainly developed further and even distributed. It's like it's parallel to what is being done actually by GeoQuery in R, except this is sort of like a Python implementation of what's being done there, and it could be developed so that I think it works as well and distributed to other people. For the loss of function, gain of function database that I put together, there's definitely certain steps that we could take in terms of validating what's there. extracting a co-expression network from it, and then combining it with interactive network to create this sort of genome control network. So yeah, again, these are potential strategies for validation. What you can do is take this library and see if you can recover the transcription factor from target lists using Enricher. So the right transcription factors come up when you put in supposing relevant targets. And then also another thing you can do is kind of the other direction where you try to recover these binding site targets by overlap analysis and say with GN and encode, which a little bit of that has been done, sort of preliminary work. But yeah.
Okay, so now I'm going to go into the validation portion of my drug GMT. So right here you'll see a table marked drug target search. So as is widely known, drugs have target portions, so target genes, which they target. That's further down. Well, one of the validations which was suggested to me was to look at how many drug targets are picked up in the differentially expressed genes for the entire GMT. And here's a table illustrating effectively how many drug targets were picked up by the various methods. So right now let's just look at the short graphs. So in the GMT, characteristic direction picked up 83 drug targets in differentially expressed throughout the entire GMT, and the t-test picked up 97. Now this would seemingly indicate that characteristic direction performed worse, but this isn't actually true, and we'll get into why that is slightly later. Only 83 targets. And just a quick reason as to why that is. The cutoff. So here we're working with a cutoff set of genes. And the cutoff is based on Ed's 4-part, increasingly conservative cutoff. Works very well for the t-test and seems to be a relatively standard method of cutting off genes with significance based on their p-values. However, with characteristic direction, as Kevin alluded to earlier, we don't have this knowledge of what sort of cutoff should work really well. So we picked up the same number of genes as were picked up with the t-test. And this just isn't fair to the characteristic direction because it's definitely a cutoff that is biased towards the t-test. But then afterwards, we used a piece of software developed, I think, also by Ed, called X2K, or expression to kinase, which finds the sort of upstream network of all of the genes in GMT. And in the upstream network, we found 171 targets for the characteristic direction and 170 for the t-test. So it's not a significant difference, but again, this is a data set which is biased pretty heavily towards the t-test. So that's just a sort of preliminary thing. Here, I plugged it into the sets to networks and then the network to canvas and generated a canvas of all the entire GMT with some clusters here of cancer drugs mostly. And that's kind of interesting, not that interesting just to look at. Look around it is obviously interesting. And just finally moving on to the confirmation that characteristic direction does actually perform much better. So here we'll see the histogram of all the targets found, and each one of these buckets is 1,000. So here we applied no cutoff whatsoever and took the entire gene list of all of the differentially expressed genes, regardless of how insignificant they are or how significant they are, and just ranked them. And then we saw where exactly we are finding the targets. And each one of these is a bucket of 1,000 gene rankings. And the significant ones are really just here, the first 1,000. Afterwards, no one really cares about the next 41,000 genes because they're just pretty much so insignificant that it's anyone's call as to which method will pick them up better. And in the first 1,000 genes, and in fact even in the first 2,000 genes, characteristic direction is clearly finding more targets.
Very good.